just like with respiration, we have this kind of overarching equation. In photosynthesis, we also have an equation. This equation, of course, is 6CO2 plus 6H2O plus energy in the form of light yields C6H12O6, which is glucose, and molecular oxygen. And what we really chose to focus on in this particular series of experiments was we focused on the pigments that are responsible for absorbing the light, and then we also analyzed what plants do with that glucose after they make it. So, in experiment 10.1, you looked at absorbance spectrums, or spectra, for these different pigments. And remember, you had spinach extract and you had carrot extract. So, you had spinach extract and you had carrot extract, and these two represent different collections of pigments. The spinach extract has what we call the chlorophylls, and it also had these other pigments that are called carotenoids, which are support pigments. The carrot, on the other hand, just had those carotenoids. So it just had those carotenoids. Now, in your book, there's an excellent figure that shows you the relative absorbance spectra of these different pigments. We actually had 4 different pigments of interest we were looking at. For the chlorophylls, we were looking at chlorophyll A, which happens to be the primary photosynthetic pigment in plants. We were also looking at chlorophyll B, which is another photosynthetic pigment in plants, that expands the range of wavelengths we can absorb. And then we were looking at the 2 carotenoids, xanthophyll and carotene. People always seem to have difficulty spelling the word xanthophyll. The sad fact of the matter is, if you really think about it, if you can remember how to spell this part of chlorophyll A or B, all you have to do is add Xanth in front of it and you've got that pigment. Because it's spelled exactly the same for those 3. And in case your instructor takes off for spelling. I don't know. I'm just saying. Preparation, make sure you can spell it. So, if we look at these, on this axis, if we start at 400 here, and we go to 700 here, and this is our absorbance. So this is, let's just say 1, and this is 0. Here's what we expect to see. Chlorophyll, go ahead and label 500 and 600 here as well. Chlorophyll A is going to absorb the most light around the violet-blue transition. And where am I going to see the lowest amount of absorbance for this pigment? Which represents what color, Sydney? Green. Excellent. So it represents green. So we see the lowest amount of absorbance at green. And then what do I see one last time when I get over here in like the orange or red range? I see one more little peak, like that. So this is chlorophyll A. Okay. Now chlorophyll B is going to expand the absorbance spectra. So with B, I'm going to see this, but it again is going to plummet when I get over towards green, and it's going to also have a peak in the A and B range. So the blue line here, that is chlorophyll B. And then finally, with my carotenoids, I'm going to see something probably like this. So the red here is my carotenoids. They're protective pigments predominantly. They absorb excessive light. Overall, the point I'm trying to make to you here is you didn't see these 3 individual graphs when you plotted or when you actually looked at your different pigments. No, instead what you saw was in this spinach, you wind up having all of these pigments. So what did the absorbance spectrum graph look like for spinach compared to these? What should it look like? It should look like a combination of all of them, right? Because in theory, you've got the chlorophyll pigments to absorb light at certain wavelengths, along with those carotenoids. And so you probably saw something like this for spinach. Follow-up question. What do you think the graph for carrot should look like? Just like the carotenoids. All that's in that particular sample are the carotenoids. So your carrot should look just like the carotenoids. Questions, concerns? Yes?
On the y-axis there, what did it go from 0 to
1.0. It's just an arbitrary value. You might have seen it go higher to like 1.5. But in essence, I'm just trying to show you where you would expect to see the highest level of absorbance. Okay. All right. So now we've established how these particular pigments absorb light. Now let's talk about another property related to these particular pigments. I want to talk about their polarity. So in 8.2, or not 8.2, I'm sorry, 10.2, we performed an experiment called chromatography. Chromatography is a procedure that separates substances based on a particular trait. And in this particular exercise, you were separating them based on polarity. So, we have chlorophyll B, chlorophyll A, xanthophyll, and carotene. If you rank them in terms of polarity, what you would find is your most polar would be chlorophyll B, and your least polar would be carotene. So that would mean chlorophyll A is less polar than chlorophyll B, but more polar than xanthophyll. And xanthophyll is less polar than chlorophyll A, but more polar than carotene. And in this exercise, what we did was we took a piece of paper that we used for chromatography. So, we're scientists, we're elaborate, we decided to call it chromatography paper. So we took this chromatography paper, and you guys rubbed a spinach leaf across it, putting these pigments, all of them, onto the paper at that point where you rub the leaf. Then what you did was you took this paper and you put it into a test tube that had a solvent that we use for this procedure, because again, we're scientists and we're elaborate. We decided to call this chromatography solvent. Okay? So you have chromatography solvent. Here in the tube. Now, here's what happens. When the solvent begins to move up the paper, because it's going to diffuse up that paper, as it hits the paper becomes polar. Okay? So the paper becomes polar. So which of these pigments is going to be the most attracted to the paper? Chlorophyll B, because it's polar and the paper is now polar. Now what this means is that B is going to have the tendency to stick to the surface of the paper. This would be the equivalent to if I were to race you somewhere, I gave you a pair of shoes that had magnets in them, and we had to run along a track that was made of metal, okay? Your shoes are more attracted to the track, and you're going to have a harder time running than I'm going. Okay? That's essentially what's happening here. So chlorophyll B sticks to the paper the most. So is it going to be able to move really far up the paper? No. And as a result, we wind up seeing chlorophyll B closest to our original pigment marking. What is going to be right above chlorophyll B? Chlorophyll A. So which pigment is actually going to move the farthest on the paper? Carotene. So carotene is going to be up towards the top. And just below it, we're going to see Xanthophyll. Okay? Questions, concerns about why this result is the way it is? Make sense? Awesome. All right, so we have one final experiment that we did in this particular lab, and that will give me just about 30 minutes to do biotech, which is what I think I might need. So let's talk the final experiment for photosynthesis. This shouldn't take that long, and then we'll jump right into biotechnology, because I'm sure you guys might have some questions pertaining to that. So let's talk starch storage. The 10.3 is all about starch storage. So, when plants make an excessive amount of glucose, or sucrose, or fructose, whatever they make, they typically wind up storing that molecule in polysaccharide form as starch. So, in this experiment, what we did was we had two different leaves. Actually, they were the same kind of leaf. They were either germanium or they were coleus. We had these leaves, and we had light, but maybe, I don't know how the prep went, but you might have actually just gotten one leaf. And if you got one leaf, then you can just take these two leaves and kind of combine them, because the principle is the same. One of these leaves was covered. So it was covered. Meaning, no light got to it. Now, if you got only a single leaf, what they probably did for the prepping of the lab is they covered half of the leaf. So half of the leaf is getting light, and the other half is not. So this leaf here is getting light. This leaf here is not getting light. So which of these two leaves is able to perform photosynthesis right now? The top or the bottom? The top one. The bottom one cannot perform photosynthesis. And that's because it doesn't have the light it needs to carry down. So the next thing you did, well, or they did to prep this for you, was they boiled both of the leaves in ethanol, thereby killing them. In addition to killing them, the ethanol removes all of the pigmentation from the leaves. So the leaves go from being green to being white. So whether it was exposed to light or not, you now have these white leaves. Then, we need to detect starch. So what is our molecule or substance of choice that helps us detect starch? Iodine. So we added iodine to these two leaves. And which leaf do I expect to see starch? The top one or the bottom? The top. Somebody tell me why I don't expect to see any starch. So this leaf has starch, and we notice that it turns that bluish black color. Why do I not expect to see any starch in this leaf here? Yes, rabble, 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 rabble. Yes. No, I think the general consensus amongst everyone who was saying something in unison at different points in time was that because this leaf is not exposed to light, it still needs to perform respiration, and as a result, it tapped into its starch reserves, breaking those starch reserves down. And because there's no more light, it can't perform glucose and make more starch.
Is it possible to have an obese tree?
No, it is not possible, at least to my knowledge, to have an obese tree. But I won't say it's impossible because there could be one out there. But I doubt there is. All right, so that's starch storage. Any questions there? All right.